Hi, my name is Brooke Oberlee, and I work in the orthopedics lab at the biomechanics department in BU with GROW. Contrary to popular belief, osteoarthritis affects more than just the cartilage surrounding your joints. It affects your entire joint. In hip osteoarthritis, the femoral head grinds against the acetabulum, bony spurs develop, and most pertinent to our research, the bone density and microstructure of the proximal femur changes. To better understand the risk of hip fracture with osteoarthritis, and to eventually establish biomarkers of osteoarthritis progression or develop preventative osteoarthritis treatments, it's vital we study these changes. The primary compressive group runs through the femoral neck, our region of interest. This area is very dense because it bears a heavy load during habitual movements such as walking. Consequently, changes to the proximal or to the primary compressive group and the femoral neck as a whole greatly impact the biomechanics of the hip and the risk of hip fracture. As new technology emerges, we are able to better quantify bone density and microstructure in the femoral neck by generating 3D images. In our lab, we use microcomputed tomography imaging to scan our samples and semi-automated image segmentation scripts generate or contour the endosteum and perosteum for us. The endosteum separates trabecular bone, this spongy and porous tissue here, perfect for bone marrow, blood vessels, and nerves, from cortical bone, this hard outer layer. And the perosteum separates the cortical bone from cartilage and other soft tissues. This contouring process allows us to isolate each region. However, manual corrections are necessary because the scripts are not as efficient at detecting signs of advanced osteoarthritis, such as trabecularization of the cortical bone, when the cortical bone becomes more porous, or the development of bone abnormalities. The problem is that whenever human intervention is required, there's inherent operator bias in the data, especially when the contours are not clear-cut, as it often is with the perosteum and endosteum. So for my research project, I conducted an inter-operator variability study to address this issue. My mentor, Sydney, and I, we executed the same segmentation analysis scripts for the same 10 femoral neck samples, and then we compared our measurements through a variety of statistical analyses to determine whether the methods were repeatable between operators, between us. My question was, were the differences between Sydney and I's results statistically significant? Except for cortical separation, we found no evidence to reject the null hypothesis that for all other bone density and microstructure measurements. That means that the differences between Sydney and I's results were not statistically significant for 14 out of 15 measurements. We speculate that cortical separation was the outlier because it is highly sensitive to both the perosteal and endosteal contours as well as the pore network analysis. So for instance, if one operator decides to include empty spaces in the perosteum, but another operator does not, then there would be a disproportionate number of empty spaces considered as potential pores. So with the caveat that operators reach a consensus on whether to include or exclude empty spaces in the perosteum prior to analysis, we conclude that the methods were repeatable between operators.